Morning, ladies and gentlemen. Welcome to day 3 of 5th ICMBB. Okay, so before we start, may I remind everyone here, we are now in Hall A. Please fill in your attendance form, which has been posted in the chat box. Also, do not forget to fill in the lucky draw for day 3. Okay, please fill in before 9:20. We will have a lucky draw session during the closing ceremony. Okay. So for today, it is my pleasure to introduce our plenary speaker, Prof. Dr. Kevin Gaston. He graduated from Leicester University with a BSc in Biological Sciences in 1986. He then completed a PhD in gene regulation at the University of Birmingham, graduating in 1989. After postdoctoral work at the Pasteur Institute in Paris and the Cancer Research UK London Institute, he was appointed as a lecturer at Bristol University in 1994. He was then promoted to senior lecturer and then reader, and he established an MSc program in biomedical sciences. He then moved to the University of Nottingham in 2018, and currently he is the Professor of Cancer Sciences and co-director of the Center for Cancer Sciences. He is also the director of a novel cancer sciences BSc degree program that he established in 2019. He has received recent grants from the Medical Research Council and the British Heart Foundation. With that, I welcome Prof. Dr. Kevin Gaston with his talk entitled PRH has a proto-oncogene with tumor suppressor function. Please welcome.
Hello. First of all, I'd like to thank the organisers for giving me the opportunity to tell you about my research work today. Today I'm going to be telling you about PRH-Hex, a proto-oncogene with tumor suppressor function. Or at least I hope that by the end of this talk, I'll have convinced you of that fact. I'd like to begin by just thanking the people who contributed to this project, because today I'm going to summarise work that's occurred over a number of years. This work is all resulting from a long-term collaboration with Professor Sheila J. Rahman, also at the University of Nottingham. And it's involved a lot of collaborators, and today I'm going to talk a little bit about some of the work that was done in collaboration with Roy Bicknell and Sebastian Altin. The work was funded by the UK Medical Research Council and also by Breast Cancer Now. And specifically, I'd like to thank 3 very talented people in the lab who did most of the work. That's Yudma Marcolino, Yusra Siddiqui, and Phil Kitchen. I'm sure you're all familiar with this textbook definition of proto-oncogenes and tumor suppressor genes. Proto-oncogenes, such as, for example, the gene MYC, can be converted into cancer-causing oncogenes by mutation or by overexpression. In contrast, tumor suppressor genes, such as P10, usually inhibit cell proliferation and tumor development, and they're often mutated or lost in cancer cells. The graph down at the bottom here illustrates that because it shows prostate cancer samples. Each bar here represents an individual patient and what happens to either the MYC or P10 gene in that patient. Red bars indicate amplification of a gene and blue bars indicate deletion of a gene. And you can see in this graph that in around 9% of prostate cancer samples, the MYC gene is amplified. So that means there's more copies of the MYC gene and more production of the MYC protein, and that's driving cell proliferation. In contrast, the blue bars show that there's deletion of the P10 gene. P10 gene is encodes a tumor suppressor, and the loss of P10 encourages those cells to form tumor cells. And you can see that in a significant number of prostate cancer samples, you have simultaneous amplification of MYC and loss of P10. And both of these events are driving tumorigenesis. So the PRH protein, or protein-rich homodomain, is also known as hematopoietically expressed homeobox. But we don't like to use that name because, as I'm going to show you in this talk, the PRH protein is expressed in multiple different cell types, and it isn't restricted to the hematopoietic compartment. So I'm going to call it PRH. PRH is a transcription factor that regulates cell proliferation and cell migration. PRH does this by controlling the expression of a whole number of different genes in the cell. Many of those genes encode growth factors and growth factor receptors. By changing the expression of those growth factors and growth factor receptors, PRH can control these processes. PRH also changes cell phenotype, and I'm going to talk in detail later about epithelial mesenchymal plasticity and the role of PRH in that. We've studied PRH in a number of different disease states, and today I'm going to be talking about prostate cancer, breast cancer, and bile duct cancer. We've also studied the function of PRH in a disease state called intimal thickening. I haven't got time to talk about that today, and if you're interested in that, you can read more about it on PubMed. I'm going to start by telling you about the role of PRH in prostate cancer. Now, as I showed you at the beginning, the MYC oncoprotein is often amplified in prostate cancer, and the PTEN tumor suppressor protein encoding gene is often deleted in prostate cancer. But if we now add in the gene that encodes PRH, or the HEX gene, you can see that HEX is often deleted in prostate cancer samples. Around 3% of prostate cancer patients show a deletion of the HEX gene. Moreover, in prostate cancer samples, the HEX gene is often hypermethylated. That means its level of methylation is increased compared to normal tissues. You can see that in the graph on the left here, where we're comparing normal and cancer methylation levels in the PRH gene in prostate cancer. And you can see that CpG methylation is increased in the cancer samples. CpG methylation is associated with the repression of gene expression, and you can see that in the graph on the right that as methylation increases, the expression of the PRH mRNA is reduced. The increased levels of PRH gene methylation result in reduced levels of PRH mRNA expression in prostate cancer samples. You can see that in this graph over on the left here, where we're comparing normal cells with cancer cells and looking at the number of PRH transcripts in those tissues. You can see that there's a significant reduction in the expression of PRH mRNA in the prostate cancer. To extend this work, Udmar Marcolino, working in my lab, looked at a microarray containing multiple prostate cancer samples and stained them with an antibody for PRH. What you can see is that as the tumors become more aggressive, and that's shown by the Gleason score here, the higher the Gleason score, the more aggressive the tumor. You can see that as the Gleason score increases, the expression of the PRH protein goes down. So what I've shown you so far is that PRH mRNA and protein levels are reduced as prostate cancer develops. So to investigate what PRH is doing in these prostate cancer cells, we turn to prostate cancer cell lines and normal prostate cells. Our model for normal prostate cells is a cell line called PNT2C2 cells. These are not quite normal because they're immortalized, but they're as normal as we can get in terms of cell biology. So we call these our normal prostate cells. And we compare those cells to prostate cancer cells, such as DE145 cells, which are a prostate cancer cell line. To look and see what PRH is doing in these cells, we can either overexpress the protein, in this case we're using an adenovirus to overexpress the protein, or we knock down the protein using shRNA. When we overexpress the PRH protein in either the normal prostate epithelial cells or the prostate cancer cells, you see a strong reduction in cell proliferation. So that's shown here as a reduction in cell number as we add the adenovirus that expresses PRH. And you can see over on the right here in this Western blot here that the MYC-PRH protein, which is a MYC-tagged version of the PRH protein, is expressed strongly in these infected cells. If we do the converse experiment and we knock down the PRH protein expression using an shRNA and then look at cell proliferation, we see that as we knock the protein down, cell proliferation goes up. And that's measured here by BRDU incorporation in this graph. And you can see the protein levels of the PRH protein are reduced when we add the shRNA. So PRH is controlling the proliferation of these cells. So I've shown you that when PRH levels are changed, it has an impact on cell proliferation. But that's not the only thing that changes when we change PRH levels. Normal epithelial cells are held together by tight interactions that are mediated by a protein called E-cadherin. E-cadherin acts as a sort of a molecular glue that sticks epithelial cells together to form tissues. But those cells can transition into mesenchymal cells, which have a completely different phenotype. Mesenchymal cells have the ability to migrate, and they will move through other tissues, and they express low levels of E-cadherin, the protein that would normally hold the cells together. And they express high levels of a protein called fermentin, which is a marker for mesenchymal cells. When PRH levels change in these cells, it changes this mesenchymal to epithelial plasticity. When we knock down PRH in these normal immortalized prostate epithelial cells, they begin to migrate more rapidly. And we can measure that easily using something called a chemotaxis assay, which is shown in the graph on the left here. When we knock down PRH in these PNT2 cells, they begin to migrate towards a chemical stimulus. And you can see that in the Western blot that the PRH levels are reduced in the knockdown cells. Conversely, when we overexpress PRH in prostate cancer cells, which have already gone through this transition and which are already mesenchymal, we can reduce the ability of these cells to migrate. This is shown in an invasion assay here, where we're looking at the ability of cells to invade an extracellular matrix. And you can see that when we overexpress PRH in these cancer cells, either PC3 cells or DU145 cells, when we overexpress PRH in these cells, it blocks their ability to invade this extracellular matrix. So PRH is also controlling this EMT changes that occur in prostate cancer cells. So to study what PRH does in more detail, we turn to looking at RNA-seq and other genome-wide measures of protein activity. And we looked in PC3 cells because these are the prostate cancer cells that I mentioned earlier. What we wanted to be able to do is to turn on the expression of PRH in these cells. To study that, we made a doxycycline-inducible expression system that would work in PC3 cells. So in these cells, we have a MIG-tag PRH construct. When we add doxycycline, we see the production of the MIG-tag protein, as you can see in the Western on the left here. Now, we have to be careful here because the doxycycline itself will have an effect on the cells. So if you can see in these cell survival graphs over here on the right, you can see that if we add very high concentrations of doxycycline, it does inhibit the growth of PC3 cells. But if we look at our PC3 PRH lentivirally transduced cells, you can see there that even when we go to very low concentrations of doxycycline, that would have no effect on the PC3 cells. This low concentrations of doxycycline will completely inhibit the growth of the PC3 cells. So we took these PC3 cells and the PC3 PRH cells that are expressing PRH, and we extracted mRNA and then did RNA-seq analysis. I've just shown in the graph down at the bottom here some of the genes that we've looked at in that analysis. So for example, the HX gene, which encodes PRH, you can see that that is upregulated as you'd expect because we've turned on the expression of PRH. The CDH1 gene, which encodes E-cadherin, is also increased in its expression, as you might expect, because these cells are becoming more epithelial. And also TGF-beta2 and TGF-beta-R2, which are the TGF-beta ligand and TGF-beta receptor genes, they're downregulated by PRH overexpression. So that's looking at a few specific examples, but by RNA-seq we look at all of the genes that are present within the cell. When we do that, we can see that in the PC3 cells, doxycycline treatment will change the expression of about 300 genes. Whereas in PC3 PRH lentivirally transduced cells, doxycycline will change the expression of about 1100 genes. So we take away the genes that are regulated in PC3 cells alone by doxycycline, and we just analyze the genes that are regulated by doxycycline specifically in the PRH expressing cells. And when we do this, we can then analyze which gene sets are being changed by PRH overexpression in these cells. And perhaps not surprisingly, when we do that and we look at the gene sets that are changing in their expression, the ones that are changing the most are the sort of things that we would expect to see change. So we see very strong changes in the E2F target genes, G2M checkpoint genes, and MYC target genes in these cells. And these gene sets, which are highlighted in red at the bottom here in this graph on the left, are all involved in cell proliferation. But we also see the epithelial mesenchymal transition genes that are changing as well. So this RNA-seq analysis sort of confirms what we see in our biology, which is that PRH is regulating the proliferation of prostate cancer cells, and it's also regulating their ability to migrate and to invade, which are both properties of cancer cells. So I'm going to change topic now and talk about the role of PRH in breast cancer progression. It turns out that the PRH protein has low expression in some breast cancer subtypes, specifically in basal cell and HER2-enriched breast cancer. So the graph on the left here shows PRH mRNA expression in tissue samples, and you can see that there's lower expression in basal and HER2-enriched. And that low expression also correlates with poor survival. So in breast cancer, in the graph on the right, which is the Kaplan-Meier analysis of survival versus PRH expression, in black we have low PRH expression and red high PRH expression. And you can see that low PRH expression correlates very strongly with reduced patient survival. So we know that PRH expression seems to be decreasing in some types of breast cancer. So to study this in more detail, we took some normal breast cancer tissues, some low-grade DCISs or ductal carcinoma in situ, and some carcinomas, and looked at PRH expression levels using specific antibodies. Surprisingly, what we found was that as we go from normal tissues and progress into cancer tissues, PRH nuclear expression is decreased. So it's not so much that there's less PRH protein around, it's that there's less PRH protein within the cell nucleus. And you can see that on the left here in these pictures. You can see that in normal cells when we're staining for PRH, you can see that there's good nuclear staining, whereas in DCIS and in invasive carcinoma, there's much less nuclear staining. So PRH protein levels are being changed, but not necessarily in the way that they are in prostate cancer samples. There's a slight difference here between prostate cancer and breast cancer, but in both, PRH nuclear activity is being reduced. So having seen that PRH protein levels are being reduced in the nucleus of breast cancer cells as they go towards being carcinomas, we decided to look and see is the PRH protein doing the same things that it does in prostate cancer cells. So we used our two standard approaches that I showed you earlier. We either overexpress the protein or knock it down using shRNA. When we overexpress the PRH protein in breast cancer cells, it inhibits their proliferation, as you can see in the graph here on the top right. When we knock down the PRH protein, as shown on the top left, then we see an increase in the proliferation of the cells. So PRH seems to be negatively regulating breast cell proliferation. And we can also measure that using things like BRDU incorporation, where in the knockdown cells have increased proliferation. And also if we analyze the cell cycle, we can see that we have more cells in S phase. And so we're regulating the proliferation of these cells. So I've shown you that the PRH protein levels in the nucleus of breast cancer cells is decreased as they become carcinomas. And I've also shown you that PRH is a negative regulator of breast cancer cell proliferation. So of course we wanted to know what would happen in an animal model of breast cancer if we were to overexpress the PRH protein in breast cancer cells. To study that, we worked with Joe Ragg and Roy Bicknell, who I mentioned at the beginning, and we look to see what would happen is if we overexpress PRH in 4T1 cells. 4T1 cells are a mouse mammary cancer model that can be, these cells can be injected into mice, into the fat pad, and they will form tumors. If we inject 4T1 cells into these mice, we see a rapid induction of tumor formation and tumor growth, shown by the white boxes in the graph on the left. But if we overexpress PRH in these cells, we see a strong reduction in tumor growth. We also should see a reduction in the number of cancer cells that are present within the liver, lungs, and spleen of these mice. The reduced presence of the cancer cells in the organs of these mice made us think that PRH overexpression might also be inhibiting the metastatic spread of cancer cells. To look at that in more detail, we collaborated with Sebastian Altin and Ling Li to look at what would happen if we injected these 41 cells into mice and then looked at lung colonization. So in this experiment, the cancer cells are injected behind the eye of the recipient mice, and then we measure the presence of luciferase labeled 41 cells in the lungs of the injected mice. And what you can see here in this experiment is that in cells when cells are overexpressing PRH, we see a reduction in lung colonization. So there's less labeled cells present within the lungs of the mice. So this suggests that PRH overexpression is inhibiting the metastasis of these cells as well as their ability to form tumors. So far I've shown you that the PRH protein appears to act as a tumor suppressor in breast cancer cells and in prostate cancer cells. What I want to do for the last 10 minutes of this talk is focus on the role of PRH in a different type of tumor altogether, which is cholangiocarcinoma or bile duct cancer. This work was a collaboration with Phil Kitchen and Sheila J. Rahman at the University of Nottingham. We were particularly interested in cholangiocarcinoma because we noticed that when we look at publicly accessible databases, there's very high level expression of the PRH mRNA in cholangiocarcinoma cells. That's indicated by the blue arrow here on the left. So Phil decided to look at some cholangiocarcinoma tissues for PRH expression, and he compared the expression of PRH in bile ducts and in bile duct cancer cells. And Phil found that in bile duct cancer cells, you had a higher protein staining for PRH in the bile duct cancer samples. So normal bile ducts have very low expression of PRH, whereas bile duct cancer cells have very high expression of PRH. So of course we wanted to know, what is the PRH protein doing in these bile duct cells when they become bile duct cancer cells? And it turns out, and I'm going to show you, that the PRH protein acts as an oncoprotein in bile duct cells. If we look at PRH expression in either primary cholangiocytes, so these are normal cells, you can see there's very low PRH expression as shown in the western on the left. But in bile duct cancer cells, which are either from a European bile duct cancer cell lines or Thai cancer cell lines, because cholangiocarcinoma is a particular problem in Thailand, so we study Thai samples of cholangiocarcinoma, and you can see that both of these types of cholangiocarcinoma have high level expression of the PRH protein, whereas the normal cholangiocytes have very low expression, if any at all. And if we look at immortalized cholangiocytes, these AKM1 cells shown on the end of this western blot, you can see that they don't have any expression of PRH. So PRH is expressed at very low levels in normal cholangiocytes, but at very high levels in prostate cancer cells. So we wondered what would happen if we took some prostate cancer cells and injected them into a xenograft mouse model and then knocked down the PRH protein to see what would happen. If we inject the cholangiocarcinoma cells into these mice, you can see that they form tumors. That's the blue line in the central graph here. So these are the control cells. So in this particular experiment, in 9 out of 9 mice, we form tumors when we inject these cholangiocarcinoma cells into the mice. But if we take PRH knockdown cells and inject those into the mice, then they fail to form tumors, or if they do form tumors, the tumors grow very slowly. So only 3 out of 9 mice in this experiment formed tumors, and when they did form, they didn't grow very much. We can also see that in human cells. For that experiment, which is shown on the right here, we took some primary human biliary epithelial cells from human donor livers. So these are cells that have been extracted from normal liver samples. We take those normal liver cells and we grow them in soft agar. And of course, because they're normal cells, they refuse to grow in the soft agar conditions. So when these are called BECs, or biliary epithelial cells. We put the BECs into soft agar, we don't see any colonies being formed. But if we overexpress the PRH protein in these normal biliary epithelial cells, or BECs, now we see the formation of colonies in soft agar. So in soft agar, these cells can grow. That indicates that the PRH protein is acting with oncoprotein properties in these cells. So what about cell migration and cell invasion? Well, as you might expect, the opposite happens in these cells to what we see happening in breast cancer cells and prostate cancer cells. For these experiments, we took some of these biliary epithelial cells, so these are completely normal cells, or the AKN1 cells, which are the immortalized biliary epithelial cells. So both of these cell types are effectively normal cells. And then we overexpress the PRH protein in these cells using a MIG-tag PRH construct. When we do that in the primary biliary epithelial cells, you can see that the expression of E-cadherin is strongly reduced, shown in the Western plot here, and the expression of vimentin is strongly induced. So that means these cells are going through an EMT transition and the cells are becoming more mesenchymal. The effect that this has on the cells is to increase their migration and to increase their ability to invade. In a migration assay, when we overexpress PRH in the biliary epithelial cells, we see high levels of cell migration. Similarly, in an invasion assay, when we look at the ability of these cells to invade an extracellular matrix, when we overexpress PRH in the BEC cells or in the AKN1s, we see increased levels of invasion. So overexpression of PRH, as well as making these cells proliferate more rapidly, is also making them invade and migrate. To look at what the PRH protein is doing in these cells in more detail, we turn to RNA sequencing as we did before in the breast and the prostate cancer samples. And in these cells, I won't go through the details here, but the gene sets that change are the same gene sets that we see changing in the breast and prostate samples. So, for example, we have changes in the epithelial mesenchymal transition gene sets. We have when we knock down PRH, and we have changes in the MIG target genes, and again, epithelial mesenchymal transition genes when we overexpress PRH. But one particularly interesting gene set that was changed are the Notch signaling genes. And it turns out that when we alter PRH levels in these cells, we see strong regulation of Notch signaling. We think that in these cells, it's increased Notch signaling is acting as an oncoprotein focus in these cells and encouraging these cells to proliferate more and to invade and migrate. Our current work is focused on trying to understand how PRH reprograms gene expression in different cancer cell types. In this example, we're looking at genes that are differentially expressed when PRH is upregulated in cholangiocarcinoma cells, which are in blue here, compared to the differentially expressed genes in prostate cancer cells when PRH is overexpressed, which are in yellow. What you can see straight away is that there's actually very little overlap between the gene expression changes in the CCLB cells and the gene expression changes in the BC3 cells. So there's a small number of genes at the interface here which are regulated inversely in either the CCLBs and BC3s. So by trying to understand these gene sets, we're trying to figure out exactly how PRH brings about the changes that it does in these different cell types. So in conclusion, I've shown you that PRH is inactivated in prostate cancer cells and breast cancer cells via multiple mechanisms. These mechanisms can include increased gene methylation and also gene deletion, and also changes in protein localization within the cells. The normal job of PRH in these cells seems to be to regulate proliferation and migration. And in these cell types, PRH acts as a tumor suppressor. It also acts as a tumor suppressor in some types of leukemia, but I haven't had time to talk about that today. But if we look in bile duct cancer cells and also in some other types of leukemia, which again I haven't had time to talk about, PRH is overexpressed, and in these cells it acts as an oncoprotein. So PRH is encoded by a proto-oncogene with tumor suppressive function. Now, PRH hex isn't the only example of a proto-oncogene with tumor suppressor function. Around 4 years ago, Shen and co-workers published a review where they put together a list of the different genes that fall into this group. It turns out that most of these genes encode either transcription factors or protein kinases. We were particularly interested to see that Notch3 is also a member of this group because PRH regulates Notch3 and Notch3 regulates PRH expression. So finally, I'd like to thank once again the people who've contributed to this work, especially my co-investigator, Professor Sheila J. Rahman, and thank you for your attention. If you have any questions, I'll really try and answer them, but I think it might have to be by email. Thanks again. Bye-bye.
You are muted, Dr. Nazifa.
Sorry. Thank you, Prof. Kevin. Unfortunately, he's unable to be with us here today. However, he's happy to take any questions through email. His email is in the chat box. Our next speaker, Associate Prof. Dr. Pang Yafan, is here with us today. And if you have any questions for her, please drop it in the chat box. I will compile all questions and we will have a Q&A sessions at the end of the session. Dr. Pang completed her first degree in biotechnology with management at the National University of Malaysia, after which she completed her PhD in biological sciences at the National University of Singapore under the supervision of Prof. Manjunatha Kini. She also obtained her postgraduate certificate in higher education from the University of Nottingham in 2018. In 2004, Dr. Pang joined Genome Institute of Singapore as postdoc fellow. From 2006 to 2012, she worked as postdoctoral scholar and senior research fellow at the University of California at Los Angeles and Northeast Ohio Medical University, respectively. Under the mentorship of Prof. William Chillian, she and her colleagues successfully established and perfected the surgical procedure to induce coronary collateral growth in rodents that help to address the mechanisms underlying ischemic heart disease. In 2009, she received the American Heart Association Fellowship as the starting point to prepare for her independent research career. After a short stint as Senior Research Fellow at the Yong Lu Lin School of Medicine, National University of Singapore in 2012, she left for Cambridge University, where she established and simplified another delicate surgical procedure coupled with echocardiography to obtain pulse wave velocity measurement in mouse. She is well-trained in molecular biology and biochemistry techniques, and she is an expert in performing delicate surgical procedures in rodents. With that, please welcome Associate Prof. Dr. Pang Yafan with her talk entitled Profiling of the MicroRNA Landscape Within Human Vascular Smooth Muscle Cells and Exosomes During Replicative Senescence. The screen is yours, Dr. Pang.
Thanks, Dr. Nazifa. Can you see my screen and can you hear me well?
Yes, we can.
Okay, so I can start now. Okay, good morning, ladies and gentlemen, colleagues and students. First of all, I would like to thank the organizing committee for giving me the opportunity to present in this conference. So first of all, let's look at the prevalence of non-communicable diseases worldwide. Okay, so non-communicable diseases kill 41 million people each year, and that is equivalent to 7 in every 10 deaths worldwide. So there are currently 5 major groups of diseases that contributed to the most death cases under non-communicable diseases. So they are the sorry, I have some technical issue here. Okay. Okay, So they are the cardiovascular diseases, diabetes, cancer, chronic respiratory diseases, as well as mental health condition. So since I'm working on cardiovascular diseases, so let's look at the prevalence of cardiovascular diseases in Malaysia. Sorry, Dr. Nazifa. So I have some technical issue here. I'm not able to proceed to the next slide.
Oh, I think, can you unshare your screen and then reshare?
Yeah, I think probably I just pause the share. Okay, let me escape now, yeah?
Yeah, sure.
I'm not sure. Okay.
Dr. Pang, are you sharing screen or are you sharing window? Maybe you
Yeah, I think
share and then you share the screen.
yeah, I think probably I should share the screen.
Yeah. So if you can unshare first and then share the screen.
Sorry, yeah.
Okay.
I'll share, new share. Yeah, I'm not able to advance as well.
You can't, because we are still seeing your screen. Okay, now we are seeing your, okay, now it's gone. Just now we are seeing your screen.
Okay, so I share again.
Yeah. Okay, we are seeing your screen now.
But I'm not able to advance. Okay. Okay, sorry.
Okay.
Okay. Sorry for the technical issue. So now let's look at the prevalence of non-communicable diseases worldwide. So non-communicable diseases kill 41 million people each year. That is equivalent to 7 in every 10 deaths worldwide. So there are 5 major family of diseases identified under non-communicable diseases that contributed to the most death cases. So they are the cardiovascular diseases, diabetes, cancer, chronic respiratory diseases, as well as mental health condition. So since I'm working on cardiovascular diseases, so let's look at the prevalence of cardiovascular diseases in the world. So in the world, cardiovascular diseases is the #1 disease killer, but this is before the COVID pandemic, okay? So cardiovascular diseases comprises of a group of disorder of the heart as well as the vasculature. So even stroke and peripheral artery disease are part under cardiovascular diseases, okay? So cardiovascular diseases kill 17.8 million people each year worldwide, and there are a lot of risk factors that contributing to this. So for example, high blood pressure, high cholesterol, obesity and diabetes, and even unhealthy diet, air pollution, physical inactivity, the use of tobacco, smoking, kidney disease, and many others. So how about cardiovascular diseases in Malaysia? So before pandemic, you know, cardiovascular diseases are the #1 disease killer in Malaysia. So in 2019 itself, okay, ischemic heart disease killed 16,000 people, okay, in Malaysia. So, you know, we have currently 1.7 million Malaysians suffered from 3 major risk factors, and we have 3.4 million Malaysians suffered from 2 major risk factors, indicating that we need to put more effort to reduce the incidence and prevalence of cardiovascular diseases in Malaysia. So now I would like to share with you the work in my laboratory entitled Profiling of the MicroRNA Landscape within Human Vascular Smooth Muscle Cells and Exosome during Replicative Senescence. So first of all, I would like to talk about ischemic heart disease. So this figure here shows a healthy coronary blood vessel, and this one is the coronary blood vessel with atherosclerotic plaque. Okay, so atherosclerosis process is a chronic process, and it can take 20 to even 30 years for the patient to experience symptoms such as heart palpitation, chest pain, shortness of breath, dizziness, as well as loss of consciousness due to the obstruction of the blood flow in the coronary blood vessel. So when the plaque, atherosclerotic plaque, grow bigger and bigger and bigger, and over time, the region here will suffer from high blood pressure as well as turbulence flow. Okay, so over time, the plaque will become unstable and the plaque will rupture. So when the plaque rupture, it leads to a phenomena that we call thrombosis. So thrombosis in the layman term is basically blood clot, but it's actually inside the blood vessel, okay? Not on the surface of your skin, but it's actually inside the blood vessel. So blood clot will basically obstruct close to 100% of the blood flow, and it usually leads to unfavorable outcome, which is sudden death, okay? So in the clinical settings, so the clinician will try to determine whether the plaque is a stable plaque or is an unstable plaque. So if a stable plaque, so the clinician can basically, put the patient on waiting list, okay, before schedule them for operation. If it's an unstable plaque, so the clinician have to schedule them for surgical procedure probably in 2 or 3 months' time. Okay, so in my previous mentor laboratory, he has 2 teams. So the first team will work on trying to collect thousands and thousands of echocardiography images and trying to look for the features and characteristics of unstable plaque. And then he also has another group of scientists that are trying to look for the biomarker that will indicate the plaque will become unstable and it's going to rupture very soon. Okay, so before I go into my study, I would like to give you a little bit introduction on the mechanism of the atherosclerotic plaque development. It's very brief because it will take me 2 hours in my lecture. So towards the end of this atherosclerotic plaque development, right, you have the macrophages that swallow the LDL, that's the cholesterol, in the subendothelial space, okay? And then it forms foam cells. So these foam cells will secrete a lot of signaling molecule, such as cytokine involved in inflammation, as well as migration and proliferation. Okay, so this cytokine will actually promote the migration of human vascular smooth muscle cells, and they form the fibrous cap here, as shown here. So this layer is not endothelial cells, but is the migrated human vascular smooth muscle cells. Okay, so this lead to the hypothesis of my study. So approximately 10 years ago, we basically hypothesized that vascular human smooth muscle cells senescence, because the cells become senesced, become old, and therefore it will eventually die off. And this phenomena leads to plaque instability and then plaque rupture. So back then, the signature microRNA expression profile of human vascular smooth muscle cells and their exosome during replicative senescence was still unknown. So this led to the objective of our study. So the objective of our study was to perform fundamental characterization and profiling to identify the signature expression of microRNA in human vascular smooth muscle cells and exosome during replicative senescence. So these are the methods that we use in the laboratory. So first of all, we apply clinical ethics from MREC and CEREC. CEREC, basically, this is the clinical ethics from Nottingham, because initially we collected atherosclerotic plaque from cardiothoracic surgeon from Hospital Sedang. So, we need to apply MREC. And then after that, we grow the human vascular smooth muscle cells in the laboratory, and we have to characterize them to ensure they underwent the replicative senescence process. And then after that, we isolated the exosome, and we have to characterize them to ensure, our technique in isolation were correct. After that, we perform sequencing analysis, we do bioinformatics analysis, and then after that, we validate the expression. Okay, so this is another methods, the flow. So to help you in understanding what we are actually doing in the laboratory. So first of all, we have 2 populations of the cells, young and senescent cells, basically human vascular smooth muscle cells are primary cells. So this is unlike cancer cell. Okay, so it's harder to grow, and you can only passage like few times. So to ensure the cells underwent senescence, replicative senescence, we basically stain them with beta-gal staining. So the senescent cell will stain blue, okay? And then also we isolated the total RNA, and we perform qPCR on some of the cell cycle arrest marker, for example, P21, P27, as well as P16. But for your information, these two methods, SA beta-gal staining as well as the cell cycle arrest marker, these are the generic marker for senescence. So when we talk about senescence, there are two types of senescence. One is stress-induced senescence, and another type is replicative senescence. So in our case, we are actually looking at replicative senescence. So these senescence characterization are generic for senescence, but the telomere length measurement will be the classical hallmark of replicative senescence. So for your information, our telomere is actually the cap at the chromosome. So every time when cells divide, the telomere will become shorter and shorter. Okay, so this is basically the classical hallmark. And then after that, we perform. Once we ensure our cells underwent replicative senescence, then we collected the condition media. Then we spin the condition media using ultracentrifugation with sucrose gradient. We collected the exosome. To ensure we have successfully in isolating the exosome, then we perform Western blot using some of the marker, for example, TSG101 protein. And also we look at the exosome in terms of the size as well as the ultrastructure using transmission electron microscopy. Okay, and then after that, we built the library using the True-6 small RNA library prep kit from Illumina. And then after that, we send the samples for next generation sequencing. So, we paid the company to do the primary as well as the secondary analysis for us. As for the tertiary analysis, we did it in our lab. So, from the differentially expressed microRNA, right, we look at the mRNA target prediction. We look at the microRNA and mRNA interaction network, protein-protein interaction, as well as the pathway involved in the differentially expressed microRNA. We also validated our differentially expressed microRNA using qPCRs. Okay, so these are the results that we obtain. So first of all, this figure here shows the young human vascular smooth muscle cells, and it shows a very nice spindle shape and is confluent. So in the senescent human vascular smooth muscle cells, you can see the morphologies are basically enlarged, and they have very irregular shape. Okay, when we stain with SA beta-gal, you can see the senescent cells stain positive for SA beta-gal. So when we perform qPCR, cell cycle arrest marker, for example, P16, P21, P27, these are upregulated. And then when we do telomere length measurement, we observe a shorter telomere as shown here. So this indicates that we already gotten a population of human vascular smooth muscle cells that underwent replicative senescence. Okay, after that, we collected the condition media. And then we try to isolate exosome from the young as well as the old cells. Okay, as you can see from the images here, from the TEM, basically the size of our exosome isolated was around 100 nanometer. Okay, and then interestingly, the exosome from senescent human vascular smooth muscle cells show increased clustering as shown here. Okay, as compared to the young exosomes, as compared to the exosome isolated from the condition media from culturing the young cells. Okay, so when we perform the protein marker for exosome, as you can see, intracellularly for TSG101, so we gotten a band of approximately 50 kilodalton, which is the reported size for TSG101. However, for exosome, when the exosome was secreted outside the cells. So we observe a 20 kilodalton increase in size. So this increase in size probably due to post-translational modification, but until today, the post-translational modification has not been reported. Okay, so it might open up a new research area. Okay, so now we have already successfully in confirming that we already isolated the exosome. So the next step is we build the library and then we send our samples for sequencings. And this data shows our heat map. And as you can see here, the post-sequencing data consisted of 127 million reads. And from this data, after we do the sequencing, after we do the alignment, so besides microRNA, we also obtain tRNA, mRNA, mitochondria, mRNA, tRNA, as well as no RNA, snRNA, as well as link RNA. So after we filter all the unwanted noise, we basically obtain 3,806 mature microRNA. And from here, okay, only 73 microRNA were differentially expressed. Okay, so after we perform a more stringent FDR adjustment, okay, as you can see here, we basically obtain intracellularly 2 microRNA that were downregulated and 6 microRNA that were upregulated. For exosomal, basically 2 microRNA that were downregulated and 1 microRNA that were upregulated. And then we further went back, to our clinical samples, and then we basically validate the dysregulated microRNA. As you can see here, we only obtain 3 validation figure. We were not able to validate the MIR7704 because this primer has very high GC content, and therefore we were not able to design the primer. Okay, now let's look at the novel upregulated intracellular and exosomal microRNA that we identified during replicative senescence. So,
sorry, you have 2 minutes left.
Intracellularly, we observe this MIR, and they basically involved in protein phosphorylation, subcellular localization, and intracellular transport. For exosomal, we observe 7704 involved in intercellular communication. As for the downregulated intracellular and exosomal microRNA, we observe MIR1555P and MIR20A5P. And they are basically implicated in increased oxidative stress, cytoskeletal reorganization, and cell cycle arrest. For exosomal microRNA, again, we observe 1555P and 146B5P. Basically, they involve in cell cycle arrest and reduce inflammation. So, we are quite happy because this pathway, they basically involve in senescence. These are the classical hallmark of senescent cells, and the consistent appearance of MIR155 indicating that it might be a potential biomarker. So when we look on the literature, we found out that MIR155 basically involved in atherosclerosis, as well as hypertension. So we suggest that, and we propose that MIR155 might be a very important player that link atherosclerosis, hypertension, and vascular aging together. So in summary, we have identified sets of dysregulated RNA involved in this process, and we also have a lot of unknown microRNA identified from our study. So in conclusion, we are the first to profile intracellular and exosomal microRNA in human vascular smooth muscle cells. We observe exosomal clustering in the O from the exosome isolated from the O cells, as well as post-translational modification is a novel post-translational modification on TSG101. And we also identified MIR155 as a potential marker during replicative senescence. So now let's come back and wrap the whole study. Go back to revisit the hypothesis and the problem statement. So, as I told you before, because the waiting list is very long, so the clinician will have issue in determining whether the patient will have a stable plaque or unstable plaque. Okay, so hopefully our biomarker the sets of biomarker identified will help the clinician in terms of decision making. So with that, I would like to thank you for all my collaborators, funders, as well as my students. Thank you, and I can take the question towards the end.
Thank you.
Okay, sorry for the technical issue. Just
Sure,
now.
no problem. Thank you, Dr. Pang, for a very interesting talk. If you have any questions for her, please drop it in the chat box, and we will discuss with her at the end of the session. Next, we are going to have a short break for 5 minutes, and we will be back with the oral presentation.
Bioplex assay development is built on years of expertise, with a rigorous SMARTS development approach that ensures best-in-class performance. Confidence in system performance is easy with the integration of the Bioplex 200 system, software, and consumables. Engage in the immunoassay workflow you're familiar with while getting results from multiple analytes in one well with less sample.
So we will start our oral presentation. Before that, can I check whether Miss Norazirah is here? Our other speakers are all here, but may I check with Miss Norazirah? If you are here, please DM me, okay? Because I could not see Miss Norazirah in the list of participants. So we shall start with our oral presentation. So our first speaker today for the oral presentation is Associate Prof. Dr. Faranas Amini from UCSI Malaysia. Her talk would be cell migration profile of triple negative breast cancer cells treated with a combination of a PARP inhibitor and calcitriol. Dr. Faranas, the screen is yours.
Hi, everyone. Is my voice clear? Hello?
Yeah, we can hear you.
Okay. Hi, everyone. I'm Farah from UCSI, and I'm presenting the preliminary result of ongoing research by my PhD student who is trying to develop a new formulation for treating triple negative breast cancer. Breast cancer is the most common type of the cancer among women worldwide and has a mortality rate of 12.9%, which is counted as the first cause of the cancer death in women. Also in Malaysia, breast cancer is has a second highest mortality rate after lung cancer. And triple negative breast cancer is the most aggressive tumor type with a high metastasis and poor prognosis. Because for TNBC, there is no estrogen, progesterone, and HER2 receptor, the treatment options are limited. Also, there are evidence that mastectomy and lumpectomy might cause local regional recurrence of the breast cancer in the patients in a very short time after the procedure. For TNBC, a number of monotherapy has been developed, but the problem is they are frequently ineffective because they are the presence of the innate and acquired resistance. Then now a lot of interest is toward combination of 2 or more drugs, which they have effect due to the different mechanism of action. In this study, we hypothesized that combination of the talazoparib, which is a PARP inhibitor, and calcitriol, a form of the vitamin D, could significantly reduce the cell migration compared to the monotherapies. Later, I will share with you the results that shows talazoparib actually have an effect on migration. The rationale for this combination is talazoparib and calcitriol can actually have a synergistic effect since calcitriol shown to be also a PARP inhibitor, and it can counterbalance the side effect of high dose of the talazoparib alone. Also, talazoparib is reported to cause anemia in about 90% of the patients. And on the other side, research shows that calcitriol improve anemia and reduce the need for their erythropoietin. It's known that cell migration is a crucial step in cancer cell metastasis. Drugs that disturb cell migration appear to be particularly promising in the cancer treatment. And since treatment combination in our early part of this project showed a significant effect in apoptosis and cell cycle, we were keen to assess the effect of the cell migration in this treatment. Then the aim of this study was to determine the cell migration profile of 2 TNBC cell lines treated with a combination of talazoparib and calcitriol. We selected BT20 and MDA-MB468 as a representative of the TNBC cell line, and also we use MRC5, is a normal fibroblast cell line from lung. We choose this lung cancer cell line because it's been reported that 60 to 70% of the breast cancer patients who eventually die were diagnosed with the lung metastasis. Also, triple-negative breast cancer is the most likely type of the breast cancer to develop and metastasize to the lung cancer. Then the cell is seated in an E-plate. We use a real-time cell analysis system, and we monitor the cell for 72 hours to identify the anti-proliferatory effect and also to determine the IC50 using this RTCA method and also the software. For migration assay, we also use the real-time cell analysis, but we use another plate known as a SIM plate, and we use the related software to monitor the cell migration after 24 hours. The IC50 of the talazoparib for BT20 detected as 91.6 macromolar, whilst the IC50 in MDA was 1 millimolar, indicating that BT20 was about 10 times more sensitive to talazoparib compared to MDA. For calcitriol, in both cell lines, the IC50 was 10 macromolar. And when we use the IC50 of the 2 drugs that we detected for TNBC cell line to treat the lung fibroblasts, it was found that the monotherapy as well as the combined treatment has only anti-proliferatory effect on the cancer cells and not in the normal and healthy cell. For the migration assay, in BT20, the combination treatment significantly reduced the cell migration compared to the control as well as monotherapy. This is important since evidence exists that despite the anti-tumor properties, some medications can work in favor of the tumor. For instance, Doxorubicin, a commonly prescribed chemotherapy agent, can increase the risk of migration and invasion of the tumor cell through overexpression of the CXCR4 gene by affecting downstream signaling pathway. Also, in our study, talazoparib treatment increased cell migration, which is similar to another report that they used to treat 2 melanoma cell lines. And also, when they use another PARP inhibitor, niraparib, in these 2 cell lines, they found that their migration rate enhanced compared to the control group. There was no explanation in that study. Also, we could not explain, but however, this is one of the advantages of our proposed formulation, and it can suppress the effect of the talazoparib in cell migration. For MTA MB4608, all the treatments significantly reduced cell migration, indicating that different TNBC cell lines behave differently to the talazoparib treatment. The combined treatment's ability to reduce the cell migration makes this treatment a potential strategy to overcome the metastatic tendency in TNBC. Similar results also were observed in a study on the human prostate cancer cell line, when they used 2 different cell lines, and they found that the cell migration decreased, and it's also inhibited the cell migration after treatment with calcitriol. Moreover, treated colon cancer cell line with calcitriol for 24 and 48 hours also show a reduction in the cell migration by 62 and 80% respectively. A combined treatment of the talazoparib and bazadoxify on human ovarian cancer cells also show a greater inhibitory effect on cell migration compared to the talazoparib monotherapy. Sorry. In conclusion, our combined therapy enhanced the cell migration inhibitory effect in both cell lines. Once talazoparib monotherapy had an undesirable effect on some TNBC cell line migration, the combination with calcitriol is promising, and it might overcome this disadvantage. Also, due to the genomic and epigenomic differences of the TNBC, the response to this combination were varied, calling for more precise selection of the cell line to test a specific formulation in future studies. Calcitriol monotherapy might also be an adjuvant therapy to overcome metastasis in some cases. With that, thank you. This is end of my presentation.
Thank you, Dr. Faranas. You're ahead of time, short and sweet. Thank you.
Okay.
If you have any questions for Dr. Faranas, please write it in the chat box. We will compile all questions and we will ask the speakers at the end of this session. Okay, next.
Thank you.
Next speaker, I would like to invite Mr. Tan Iwe from University of Malaya. His talk would be on targeted sequencing study of cardiomyopathy-associated genes in the Malaysian population. Please welcome Mr. Tan.
All right. Thank you to the chairperson. Can you see my screen?
No, we haven't seen yours yet, but it says that you have started screen sharing.
Yeah. Wait, hold on. Can you see the screen?
Yes, now we can see your screen.
All right. Okay. So I think I will just start. Okay.
Yeah,
Morning to all participants. I'm Iwe from Department of Biomedical Science, University of Malaya. My presentation title today is Targeted Sequencing Study of Cardiomyopathy-Associated Genes in the Malaysian Population. Cardiomyopathies, or CMPs, are a diverse group of cardiac diseases due to structural and functional abnormalities of myocardium. There are many subtypes of CMP, which the 2 commonly reported subtypes are dilated cardiomyopathy DCM and hypertrophic cardiomyopathy HCM. DCM is characterized by enlargement of left or both ventricle and systolic dysfunction. On the other hand, HCM is characterized by thickening of septum and left ventricular wall, causing difficulties of blood flowing out from the heart. The prevalence of both DCM and HCM are 1 in 2,700 individuals and 1 in 500 individuals respectively. CMP are multifactorial diseases, which some common causes include infective agents, neuromuscular diseases, and genetics. Genetics play a prominent role in CMP manifestation, which constitute 25 to 50% of DCM cases and 30 to 60% of HCM cases. For the disease to manifest in individuals, a single mutation is sufficient enough, whereas multiple mutations indicate earlier disease manifestation and more severe phenotype. There are more than 50 CMP-associated genes have been reported previously affecting various components of cardiomyocyte, such as sarcomere, nucleus, and extracellular matrix. In this study, 5 CMP-associated genes were studied, which were NYH7, NYBBC3, TNT2, TPN1, and LMA. The first 4 mentioned genes form sarcomere, which are basic units for muscular contraction, whereas LMA encodes thymine, which are essential structure forming nuclear envelope of the nucleus. Genetic studies of CMP have been conducted extensively in Western countries such as UK and France. However, only few studies have been done in Asian populations such as Singapore, China, and India. Therefore, this research study is to examine the distribution of genetic variation in 5 CMP-associated genes in the Malaysian population. For the oral study methodology, a total of 105 clinical confirmed non-ischemic CMP patients were recruited from 2 centers, UMC and IJN. Phenochloroform extraction method was used to isolate genomic DNA, which concentration and purity of extracted DNA were assessed using nanophotometer. Next, long-range PCR was used to identify entire gene region of 5 study CMP-associated genes as 50 amplicons, which the amplicons were purified and pooled together. Qubit was used to check the concentration of the purified pooled amplicon to ensure sufficient DNA input for library preparation. Nextera DNA flex kit was used to prepare the NGS library, which amplicons were fragmented and tagged with adapters. After that, the prepared libraries were QC through Qubit and bioanalyzer and sequenced using Illumina MiSeq system, which FastQ file were obtained and used for bioinformatic analysis. The details of bioinformatic analysis will be discussed later in detail. For the demographic details of the study, we have collected 105 CMP samples, which 51 samples were DCM and 54 samples were HCM. Male gender predominated in both DCM and HCM group, which constituted 62.7% of DCM cases and 59.3% of HCM cases respectively. For ethnicity, Chinese ethnicity has the highest overall CMP cases, and in both study CMP subtypes, followed by Malay, Indians, and others. For bioinformatic analysis workflow, it can be separated into 2 main sections, primary analysis and secondary analysis. Primary analysis refers to the alignment and assembly of DNA fragment in providing the full genetic sequence of a sample from which genetic variants can be determined. The whole process was executed through Dragon Germline pipeline, which FastQ serves as the input file and 2 output files obtained, BAM and VCF. Several additional software were also used, such as FastQC software for quality checking of FastQ file, integrated genomic viewer for data visualization of BAM file, and SAM2DET for computing gene coverage of each sequence sample. For this study, the mean coverage for all targeted genes were 325 times, in which the gene with the highest mean coverage was LMA, followed by TPN1, TNT2, NYH7, and NYBBC3. Variants were filtered based on the following condition: coverage more than or equals 30 times, genotype quality more than or equals 30 times, and variant frequency more than 0.2. One primary analysis has been completed. The obtained VCR will perform secondary analysis, which the discovered variants were annotated and interpreted. The variants were compared and cross-checked with databases such as CleanOne and DBSNP, and in-situ prediction tools such as SIF and Polyfern were used to predict the variant effect on the affected protein. With the compiled data, variants are classified according to ICMBB variant classification system, which classified variants as pathogenic, likely pathogenic, uncertain significant, likely benign, and benign. Pathogenic variants can contribute to the development of disease, whereas benign variant does not cause disease. For results, the studies was able to identify successfully 28 variants in total among the CMP patient, which 5 variants were pathogenic, 8 were likely pathogenic variants, and 50 were variants of uncertain significance. In addition, 4 novel NYBBC3 likely pathogenic variants were discovered, which had not been reported in the DBSNP and EXAC databases before. When stratified the discovered variants according to the study genes, half of the variants were found in NYBBC3 gene, followed by NYH7, TNT2, and LMA. There was no variants were found in TPN1 gene. Moving on to the molecular consequence of discovered variants, most variants are missense variants, which were variants that have single base pair substitution, causing change in encoded amino acid. The second highest variant type were friendship variant, which include insertion or deletion that produce non-functional protein. Next, The third highest variant type was splice region and intron variant. This type of variant destroyed RNA splicing, which led to improper intron removal and caused alteration of the open reading frame. For intron variant, splice donor variant, and splice region and Siemens variant, each variant type only reported one variant respectively. Intron and splice donor variant has similar detrimental effects as splice region variant. For Siemens variant, the codon substitution do not change the encoded amino acid, but it can affect protein expression and function. In our study CMP cohort, 31 patients were found to carry at least one pathogenic variant, like pathogenic or VUS. When we stratified variant distribution according to CMP subtype, 7 mutation carriers were reported in DCM cohort, while 24 in HCM cohort. Interestingly, out of the 24 HCM patients, there were 2 HCM cases observed to carry at least one likely pathogenic variant and 1 VUS, while another 2 HCM cases were found to carry 2 VUS, which add up to the reported amount of 28 variants. In terms of mutation distribution among different ethnicities in this study, Malay had the highest frequency, followed by Chinese and Indian. For discussion, 13 pathogenic and likely pathogenic variants were observed in 4 DCM patients and 12 HCM patients. All variants were observed in 1 patient except for NYPPC325 base pair deletion variant, which was observed in 4 patients, and TNT2 deletion insertion variants in 5 HCM patients. The study CMP-associated genes are more prominent involved in the genetic etiology of HCM than DCM in the Malaysian population, in which 84.6% of the observed pathogenic and likely pathogenic variants present in NYH7 and NYPPC3 gene. For today's presentation, only 5 pathogenic variants were discussed. For NYPPC3 gene, 2 pathogenic variants were discovered. The NYPPC3 C.1504C2T is a missense variant present in exon 17 that causes a change of adenine to tryptophan. In addition, it's a founder variant reported in causing HCM in Caucasian population. This mutation affects the C3 domain of NYPPC protein, which affects the contractile regulation of cardiomyocyte and lead to HCM. On the other hand, NYPPC325 base pair deletion is a deletion variant present at intron 32 and is a common variant that in Asian population, especially South Asian descents. This mutation causes cortical dysfunction of sarcomere. For NYH7, there were also 2 pathogenic variants being discovered. The NYH7 C.1208G2A is a missense mutation in exon 13 that results in the substitution of adenine to glutamine. The mutation located at S1 domain of beta-myosin heavy chain, which can affect ATP hydrolysis and facilitation of sliding movement in actin filament. On the other hand, NYH7 C.2221G2C is a missense variant present in exon 18 that causes the change of glycine to adenine. Like previously mentioned variant, it located at S1 domain of beta-myosin heavy chain and affect cardiac muscle fiber by reducing isometric force production and velocity. Lastly, LMA reported 1 pathogenic variant, which was LMA C.568C2T, is a missense variant present in exon 3 that causes an amino acid change from adenine to tryptophan. The mutation affected helical rod domain COI1B of DNA protein, which can affect nuclear processes like DNA replication, transcription, and repair. In conclusion, a total of 13 pathogenic and likely pathogenic mutations were found among the CMP patients. The selected genes were more prominent for HCM than DCM cohort, which most variants were discovered in NYPPC3 and NYH7 genes among Malaysian CMP patients. These are my references cited for this presentation. For acknowledgement, I would like to thank my supervisors and lab members for any assistance and criticism throughout the project, not to mention that the project is funded under 2 grants, which are Frontier Research Grant and UNC Care Research Grant. Thank you for listening to my presentation.
Thank you, Mr. Tan. A very enlightening talk. If you have any question for Mr. Tan, please drop it in the chat box and we will get him to answer at the end of the session. Okay, next, I would like to invite our speaker, Ms. Norazira Matnayan from UiTM. Her talk is entitled Attenuation of MicroRNA Expression Decrease the NMDA Receptor Subunits by Prenatal Bisphenol A Exposure in the Male Sprague-Dawley Rat Hippocampus. Please welcome.
Thank you, Associate Professor Dr. Nafiza. I'll share my screen. If you can see my slide and hear my voice.
Yes, we can see your slides.
Okay. So I will start. Assalamualaikum, hi, and a very good morning to everyone, and nice to virtually meet you. I am Noazra Maknayan from Faculty of Medicine, University Technology Mara. In this presentation, I'm going to share my findings entitled Attenuation of microRNA expression, decrease the NMDA receptor subunits by prenatal bisphenol A in the male spratlily red hippocampus. So let me start by giving you some background information about my study. Epigenetics, this is where the DNA interacts with the smaller molecules within the cells. It is known as the study of mechanism that cause changes in gene expression but do not involve alteration in the DNA sequence. There are three types of action that control epigenetics mechanism, which are DNA methylation, histomodification, and also expression of microRNA. So in my study, we are going to focus on the expression of microRNA. So microRNA is actually a short non-coding RNA that has the ability to turn off the gene expression by inactivating the mRNA into translating its genetic information to proteins. So this is actually how the cells gains characteristic and function to further develop into their specialized tissue or organ. So the regulation of microRNA is a part of normal development, and it much happens during the fetus development. Focusing on our study, I will bring you to look into how the microRNA can influence the brain development. So microRNA has a critical regulatory role during the neurodevelopment from early neurogenesis, anti-synaptogenesis, as well as the maintenance of the neurofunction. However, with the influence of diets, environmental pollutants, and also medication, reportedly can give effect to the microRNA regulation during the brain development. And it is a worrisome issue as the microRNA modification during the pregnancy reportedly can increase the susceptibility of the baby to have the neurodevelopmental disorder. And in fact, the imprint will pass the epigenetic traits until reach adulthood to the next generation. So in relation to our study, environmental pollutants such as bisphenol A, or we call as BPA, has been shown to disrupt the neurodevelopmental process, which is, this is based on the previous human and animal studies. So what is actually bisphenol A? So bisphenol A is actually an industrial chemical primarily used in the production of polycarbonate plastics and also epoxy resins. And it can be found in various consumer products such as in the plastic bottles, coatings of foods and beverages cans, toy items, and many more. So BPA in applications is highly demanded due to its rigidity and flexibility to the materials. And in fact, the demand of the BPA is increasing year by year, where it is estimated that 10.6 million metric tons of BPA production will be in the 2022. And it is an issue when a previous researcher found that heat can cause the BPA molecule to be leached from the packaging of plastics or resins into food and drinks, making the human to be more vulnerable towards BPA in our daily life. And in addition, the safe dosage of BPA, which is the no observed adverse effect level, NOAEL, at 5 mg per kg per day, reportedly can lead to the various human health issues, including to the neurodevelopmental disorder. Specifically in our study, we do a research on this on the effects of BPA towards pregnant mother, or the effects of the prenatal BPA exposure. And next, we want to see how it can influence the gene expression in the brain during the brain development. And to be more specific, when the pregnant mother were already exposed with BPA, either through foods or drinks, the BPA will be absorbed into the blood vessel and transported into the liver to be metabolized. And lastly, it should be excreted out into the urine. However, in case of pregnant mother, the BPA molecule can possibly transfer to the baby inside the womb via the placenta. So as we know, baby has an immature liver. So this will lead to the limitation of the metabolization of the BPA inside the liver, and next leading to the bioaccumulation of the BPA. So the estrogenic BPA reportedly can disrupt the gene expression, for example, to the baby's brain, which will lead to the impairment of learning and memory function when the baby reaches a later age. So due to those concerns, we do an investigation of the prenatal BPA effects on the specific part of the brain called the hippocampus, which is responsible in learning and memory. We found that in the hippocampus, there is an NMDA receptor subunits, which are GLUN-N2A and GLUN-N2B, that has been identified to regulate the excitatory synaptic transmission during the learning and memory process. And to be more specific, there are actually two types of microRNA, which are MRI19A and MRI539, that are responsible in regulate the development of the NMDA receptor subunits by targeting its mRNA. So MRI19A will regulate the development of GLUN-N2A, and MRI539 will regulate the development of GLUN-N2B. However, we have lack of information on how the interruption of the BPA towards the microRNA expression will influence the development of the NMDA receptor subunits in the hippocampus. So this brings us to the problem statement of the study, where retrospective study has shown that children with learning disabilities were exposed to the maternal BPA during fetal life. It has been suggested that prenatal BPA exposure at control dosage involved with permanent modification of NMDA receptor when reaching adulthood. However, the effects of prenatal BPA exposure at NOEL 5 mg per kg per day on the expression of microRNA and the level of NMDA receptor subunits in the male fetus and adolescents rat hippocampus have not been adequately studied yet. So we hypothesize that the prenatal BPA exposure changed the expression of MRI19A and MRI539 and its complementary NMDA receptor subunits, which are GLUN-N2A and GLUN-N2B in the fetal and adolescent male rat hippocampus. So the objective of this study is to investigate the effects of prenatal BPA exposure on the expression of microRNA and NMDA receptor subunits in the male fetal and adolescent rat hippocampus. Now moving on to the methodology of the study. So in this study, we are using pregnant Spratt-Dawley rats where they were divided into two groups. We have control and treatment groups. So the control groups, the mother were orally exposed with 0.5% of 280 in RO water, while the treated groups, the mother was orally exposed with BPA dosage at 5 mg per kg per day with 0.5% of 280 in RO water. So the male fetuses and the male pups from each of the mother will be divided into two age, where we have the male fetuses at age gestation day 21 and also the male pups at age of 35 day, adolescence day 35. So when the male pups and the male fetuses reach at that respective age, they were sacrificed and their hippocampus was collected to determine the level of microRNA and also GLUN-N2 protein by the respected parameters stated on the slide. And now moving our attention now to the results. So firstly, let's take a look at the expression of microRNA, MRI19A and MRI539. So according to the result, it showed that BPA dosage at 5 mg per kg per day downregulate the expression of MRI19A and MRI539 in the male rat hippocampus. Both stage of ages show that gestation day 21 and adolescence day 35 have a decrement in expression when we compare to the control groups. Especially for the MRI539, it shows there is a significant difference between the groups of ages. And in relation to the...
Sorry, you have 2 minutes left.
Okay, in the relation of microRNA expression, interestingly, the level of GLUN-N2A and GLUN-N2B also shown to be disrupted, and where the BPA dosage at 5 mg per kg per day significantly reduced the level of GLUN-N2A and GLUN-N2B in the male rat hippocampus. So the result revealed that both stage of ages, gestation day 21 and adolescents day 35 have with the BPA rate have the significantly lower level of GLUN2 proteins when we compare to the control group. This, as a conclusion, the prenatal BPA exposure at 5 mg per kg per day affect the expression of MRI19A and also MRI539 and its complementary NMDA receptor subunits, which are GLUN2A and GLUN2B level from fetus until adolescence age. And we can observe that the attenuation of microRNA expression and NMDA receptor subunits level lead to the persistent changes from fetus until adolescence age. And this finding suggested that the modification in epigenetic traits may inhibit the synaptic plasticity in the hippocampus, which is involved with the learning and memory function in later life. And last but not least, this finding will contribute a novel knowledge where a specific altered microRNA can be the marker for the early recognition of the offspring that has a risk of learning disorder when reaching adulthood. And at the same time, we hope this finding can lead the certain agencies or the government to propose a better risk management in BPA applications so that we can improve a healthy living environment for humans, especially for pregnant mothers. I think that's all from me. With that, I thank you.
Thank you, Ms. Norazira. Next, we have our speaker, Mr. Muhammad Ashraf Abdul Jalil from IIUM. I'm thinking about food already, looking at the title, Association between DNA methylation of the CUB and sushi multiple domains, one gene and schizophrenia. Please welcome.
Thank you, Dr. Nazifa. Can I share my screen? Because I think I cannot because...
Norazira, can you unshare? Okay, thank you. Yeah, please.
Give me a second. Okay, Assalamualaikum and good morning to everyone. Hope everyone can see my screen now. Yes, I'm Muhammad Ashraf Abdul Jalil from IIUM. I'll be presenting one of the research in the team for looking into genetics of schizophrenia. So the title is Association between DNA methylation of the CUB and sushi multiple domains, one gene and schizophrenia, although it has nothing to do with sushi. So schizophrenia is a low prevalent mental illness, which is very disabling and has a very high disease burden. Part of the reason because its age of is in the early adult years, which can be considered among the productive years of human life. So many patients have to drop out from their studies or might lose their jobs and also have problems with relationship. In fact, one study estimated that 80% of them remain unemployed and have to be dependent on their family members. So we lost a lot of money from one disease alone. So schizophrenia has a wide range of symptoms and usually divided into three categories: the positive, negative, and cognitive symptoms. And these could probably have arisen from different pathologies inside the brain. And this is why the antipsychotics, the main treatment for schizophrenia, are only effective in treating positive symptoms because they are mainly dopamine antagonists and they don't work really well with negative and cognitive symptoms. Because of this, we need to expand our understanding on schizophrenia pathogenesis by looking at other systems. So in fact, there are many evidence that have shown that other systems have also been involved in schizophrenia, including neurodevelopment, defective neurodevelopment, dysregulated immune system, genetic variation, also environmental factors. And one of the theories that have been circulating in the past decade is on synapse pruning. So I think this probably been touched a bit by the previous speaker. Synapse pruning is essentially a developmental process of targeted synapse elimination as we were growing from late fetal life until young adulthood. So it's a process to refine the brain networks, and this is performed by the immune system. So the immune cells of the central nervous system, the macrophage, also the complement system, surprisingly. So the complement system is usually involved in clearing pathogens and also after the inflammation everywhere else in the body, but inside the brain, it has a role in synapse pruning. In fact, schizophrenia brain has been found to have less synapse compared to healthy brains. Synapse pruning is interesting because it can relate many theories of schizophrenia. It is essentially from the immune system, and in fact, it affects neurodevelopment by causing abnormal synapse development, and this in turn can affect the neurochemistry balance, and this results in schizophrenia symptoms. And we know that immune system can be influenced by genetic variation as well as the environment. So CSMD1, or the CUB and Sushi multiple domain. So what it is? So in short, it's CSMD1. It's a large gene on chromosome 8. It was found as a tumor suppressor gene at the beginning, but because it has multiple domains which can act as complement inhibitors, these domains looks like a sushi in the molecular structure. So it's named so. And this protein has been found to mainly be expressed in the brain and also testes. So it's quite specific. And later research has also found that it in fact inhibits some of the complement factors or proteins. So CSMD1 has been implicated in schizophrenia by various evidence. Firstly, from the genome-wide assessment study, or GWAS, which this very large study involving 100,000 of patients, and it has been repeated twice, and both has found the same SNP variants of CSMD to be associated with schizophrenia. And then further study has found that it's associated with cognitive dysfunction, which is a phenotype of schizophrenia. And expression study has also found there's a lower level of CSMD1 in schizophrenia. In animal studies found that deletion of this gene leads to altered behavior similar to schizophrenia. And in vitro studies using neurons from stem cells have found that it has a role as a complement inhibitor. So it's probably have a protective effect against schizophrenia, because we suspect that in schizophrenia there is excessive synaptic pruning. So by being able to inhibit complement activation, it should serve as a protective factor for schizophrenia. Unfortunately, despite all the evidence, there is still no report on DNA methylation of this gene. So I think previous speaker has spoken a bit about epigenetics. So one of the mechanisms of epigenetics is DNA methylation, which is the most studied and well characterized mechanism of epigenetics. So it is addition of a methyl group to the C nucleotide, which is next to a G nucleotide. So it affects transcription, usually by repressing transcription. And also what is important about DNA methylation is two features. First, it is influenced by the environment. So it can relate the environmental factor and genetic factors together and explain the pathogenesis of schizophrenia. Secondly, it is modifiable using drugs. So it could be a potential treatment target for the future. So we want to know whether CSMD1 methylation affect its expression and subsequently affect schizophrenia as well as the psychopathology. What we mean by psychopathology is the characterization of the symptoms, because schizophrenia is a wide and heterogeneous disease. So many symptoms which sometimes looks opposite to each other. So we want to know which symptom types is actually more affected by this pathology of CSMD1. So we perform a comparative cross-sectional study involving nearly 400 subjects. And for patients, we examine them with two psychopathological scales, the PANSS and PSP, more on that later. And then for both groups, we took their blood, and from there we took their DNA, and we performed this methylite assay or qPCR assay on the gene. So it's a big gene. So how do we decide so where to look for this methylation site? So there are many methylations, possible methylation site, but where it is the CPG is found in high frequency is called the CPG island. So this is more likely where it is more influential. So we found at least many of these, but I think because it's early in the gene, so it's probably the one that is more influential in terms of transcription. So we choose this site to examine the methylation levels. For PANSS and PSP, so PANSS is actually positive and negative symptom scales, or it is actually a widely used scales to measure treatment response for schizophrenia. So in short, the higher the score, the worse the symptoms. The opposite is the PSP, the personal and social performance scales, which measure the functions of the patient. So the higher the score, the better the functions. So what have we found in terms of the social demographic? There are no difference no significant difference in the age, gender, and ethnicity of the patient, of the subjects, because we match them. But in terms of the DNA methylation, we found that there is a lower methylation level. I mean, the overall distribution is lower levels of CSMD1 DNA methylation in patients compared to healthy controls. So this is the first study to report methylation of CSMD1 gene, because in previous EUA study or epigenome-wide assessment study has found that the closest methylation site is actually about 700,000 base pair away from this gene. But it has been found in cancer studies. High methylation levels of CSMB1 has been reported in several types of malignancy, like colorectal cancers, and this methylation causes silencing of the gene, like many other genes, when the high methylation will cause repression of transcription. This is unexpected because we thought that because CSMB1 likely to be a protective against schizophrenia, we were also expecting there's a high methylation levels of this gene in schizophrenia, but instead we found the opposite. However, the methylation can also cause increase in depression as highlighted in other studies. Therefore, it could be depend on the locus and also the context. So it depends on which sites that is being methylated. So we go down deeper into the psychopathology or the symptoms. We found no correlation between the severity of the symptoms as well as the functions of the patient with this methylation in CSMB1. However, when we look into individual symptoms types, we found a weak correlation, but significant in the positive symptoms type, but not in other symptoms type. So what we can say about this is probably this pathology involving CSMB1 and synapse pruning is more related to the positive symptoms rather than the negative and as I mentioned earlier. And this is interesting because different studies have found different association about CSMB1. For example, the study on by El Gayed found that it's associated with risk of psychosis, which is part of the positive symptom. Other studies have found CSMB1 to be related with the cognitive dysfunction of the cognitive symptoms. And in animal studies, they found that it's actually related to the negative symptoms. So there's no conclusive evidence about this, so more studies are needed. And then we look also into the social demographic factors that could also influence the methylation, as we know that the methylation can be influenced by the environment. We found that age actually correlates positively with CSMB1 methylation, and there's a higher rate of methylation of CSMB1 in Malays compared to Chinese. This is unsurprising, but this is new information. But we know that these factors have been associated with DNA methylation in the past for other genes. However, it's still inconclusive, so more research is needed, and especially experimental and also mechanism studies. Since methylation is reversible, so we need to have this further exploration because it could be useful in designing a future treatment. The study has some limitations because we only measure in the blood, so it may not reflect the methylation in the brain. However, we have evidence to say that for just some genes, the methylation are similar in between blood and brain, and we couldn't test the expression of CSMB1, I think because of very, very low amount of transcript inside the peripheral blood. And the methylation of CPG islands in other CPG sites in CSMB1 was not studied, as well as in terms of other genes that could be related to the synapse pruning theory. And lastly, that our patients are mostly stable and on medication, so we couldn't really tell the difference if this is higher or lower in patients with high methylation levels. So in conclusion, we found that lower DNA methylation could be involved in the pathology of schizophrenia, particularly in the positive symptoms. However, the cause-effect relationship and the exact mechanism must be studied in future studies. So these are the reference. With that, I thank you.
Thank you, Mr. Ashraf. If you have any questions for him, please drop the questions in the chat box. And we move on to our last speaker. We have Mr. Derek Shelby Simon from University Malaysia Sabah with the talk entitled Potential of Reverse Transcription Loop Mediated Isothermal Amplification as a Simple Rapid Molecular Diagnostic Tool to Detect SARS-CoV Nucleocapsid Gene. Please welcome. Mr. Darish, can you share your slides?
Hi, can you see my slides?
Yes, we can. So if you can put it on the slideshow.
All right. Okay.
You're good to go.
All right. So good morning to the organizers of ICMBB and the participants and all present here today. I'm Derek Shelby Simon from the Biotechnology Research Institute of University Malaysia Sabah. And today I'll be presenting on the potential of reverse transcription loop-mediated isothermal amplification, RT-LAM, as a simple, rapid molecular diagnostic tool to detect SARS-CoV-2 nucleocapsid gene. So we are well aware that COVID-19 is very infectious. What's even more troubling is that its viral genome is constantly mutating. and new variants are emerging by the minute. The problem here lies when there are mutations at the primer binding sites for the current PCR test, which evades detection. So to prevent false negatives, primers must be designed from conserved regions and or to detect multiple regions at the same time. Currently, the two most common diagnostic tools for COVID-19 is the PCR or the rapid test. Briefly, PCR detects genetic materials by amplification, thus detecting even the minute amount of RNA. This is effective in detection even before the onset of symptoms. RTKs, however, it detects the antigens, which requires a higher viral load. This increases the chances of false negatives. It seems like PCR is the clear winner, right? But it is laborious, requires technical expertise, time-consuming, and most problematic of all, it's most expensive. This begs the question, how about RT-LAM? It is fast, accurate, it's also sensitive. It's previously used for SARS and MERS. But what exactly is LAM? In the most basic of definitions, it is a method to exponentially amplify the specific genome region at isothermal conditions. It utilizes 4 primers targeting 6 regions of the target gene. Then later on, it was discovered that by the additional loop primers, the amplification rate further increases. What's great is that since it is at isothermal conditions, expensive machineries are no longer required. Just like any nucleic acid amplification, the detection can be visualized by electrophoresis or colorimetric, and also by lateral flow dipsticks. So for agarose gel electrophoresis, positive results can be observed by the production of ladder-like bands. And for negative results, there are either no bands or only a single band, which are most probably the primerdimers. So the problem with this is it is also time consuming, and also it requires toxic chemical stains. For colorimetric method, using cybergreen as an example, positive results can be observed by color changes from orange to green, and negative results will have no changes. The problem here also is that at low concentration, the color changes may not be that obvious, which leads to observational errors. Here on the right, we can observe a lateral flow dipstick. Positive results can be observed by the production of distinct lines at the control and also the test lines, and negative results will only have a distinct line on the control line. The test line, the potential of this is that there can be multiple test lines at the same time, therefore reducing the chances of false negatives. So the aim of this study is to optimize a LAM protocol for SARS-CoV-2 nucleic acid regions for rapid, sensitive, and specific detection on a single LFD with multiple test lines. So to do this, LAM primers were designed based on the conserved region screened using a multiple alignment program, and then the sequence was input into the Primer Explorer program to generate the primers. From there, we optimized the LAM protocol to obtain the highest reaction rate. Once the protocol is established, the specificity and sensitivity tests were conducted. The specificity test was done by testing the assay against other closely related coronaviruses. The sensitivity test is done by testing on serial diluted positive controls. Then, once that is well established, the primers redesigned with labeled inner primers and then tested on an LFD. Moving on to the results and discussion. So we identified two conserved regions on the nucleocapsid gene after screening 165 genomes, which we include all the variants of interest and all the variants of concern. So a set of 6 primers were designed for each of these regions. So we have 2 sets of primers. Then the protocol was optimized by testing with positive control plasmid, which contains the cDNA of a SARS-CoV-2 nucleocapsid gene. And from there, we also synthesized an RNA for the same gene using T7 RNA polymerase to optimize the RT-LAM protocol. We found that a ratio of 4:1:4 of inner, outer, and loop primers were able to produce ladder-like bands from as little as 10 minutes with both sets of primers. So to test for its specificity, the primers were tested against plasmids containing the nucleocapsid gene of SARS-CoV and also MERS-CoV. As observed here, for both regions, only the SARS-CoV-2 N gene control plasmids were amplified. So for sensitivity tests, the LAM assays were conducted with both sets of primers. We were able to detect as low as 10 to the power 2 or 100 copies of the control plasmids. So what's interesting here is that when we compare it to the qPCR, the qPCR was also able to detect 100 copies of control plasmid at 40 cycles. So we can say here that the LAM protocol is comparable to that of the gold standard qPCR. Okay. So here we can see that the positive results here are on agarose gel and also cybergreen on the left. Yes, indeed, both can observe the positive results quite clearly, but it is also worth noting that for when we are using cybergreen under normal conditions, under normal lighting, it's actually quite difficult to differentiate positive to negative, especially here at 100 copies. So, and also we can observe that on the left, the results are shown separated. So for each region, we have to see it on its own gel, on its own tube, while on the right here, on the LFD, we can observe the 2 test lines on a single strip. So the potential here is that more test lines can be added to further avoid the chances of false negatives. So in conclusion, the current RT-LAM protocol is able to detect SARS-CoV-2 nucleic acid as fast as 10 minutes. And then given more time, at 30 minutes, it is sensitive up to as little as 100 copies of plasmid, which is comparable to the gold standard qPCR. It is also specific towards only SARS-CoV-2. Therefore, RT-LAM LFD is potentially a simple yet specific and sensitive method of rapid molecular diagnostics of COVID-19, with further potential to add additional test regions. So here are my references for this study. And finally, I would like to thank UMS for the funds and also the Biotech Research Institute of UMS, as well as my supervisors. Thank you.
Okay, thank you, Mr. Derek. We have come to the end of the talk. So we have, for the Q&A sessions, we have two questions for Dr. Pang. So Dr. Pang, you are here, right? Okay, I can see you now. The first question comes from Dr. Darmani. May I know how much SA beta-gal stem cells percentage activity considered as undergone replicative senescence? That's the first question. Do you want to answer that first before we go to the next one?
Actually, SA stands for senescence associated, and then beta galactosidase stain. So this stain is a generic stain for both replicative senescence as well as stress-induced senescence. And actually, we perform the staining at the acidic pH. Like pH 6. Okay, the pH is very important. So because in my previous mentor laboratory, so we have a senescence group. So this assay is something like a quick assay in which everybody will know how to do. So it's a very quick qualitative assay for cell culture as well as for animal tissue section. So all of us will know how to do it. So if you want to look at the percentage, the activity, I think possibly, but we have not do it. I think, you know, we can lyse the cells and then collect the lysates, and then we have to modify a little bit on the staining reagent, and then we can read on the wavelength to see, you know, the change in color. Then we can quantitate it. But as I said, you know, we use it as a very quick assay for qualitative purposes. And then, of course, we still have qPCR and other methods, you know, to basically differentiate between replicative senescence as well as stress-induced senescence. Okay, so I hope I answer your question, Dr. Namani.
Yes, because I just wanted to know was like, because sometimes people say how much whole cells you have, kind of thing. So do you look like how much
usually,
thing,
usually about, because the old cells, right, they will never go confluent, because, you know, after a certain passage, right, they will only like in one plate, they will probably about 20% of the cells like in one plate, because they will never divide again. Okay, they cannot divide. So usually almost all the cells will stain blue, I would say. You know, if there is cells, they will stain blue because they cannot divide anymore.
Okay, thank you.
Okay, so next question from Dr. Darmani, directed to Dr. Pang as well. Do you think HSA-MIR1555P is also an important biomarker for stress-induced senescence?
Yeah, I think so, because just now when I look at this question, right, I just went to PubMed and search. Actually, MIR155 has also been implicated in stress-induced senescence in fibroblasts as well as microglia cells. But in these cells, human vascular smooth muscle cells, I myself have not done it, you know, because, you know, you need to create a stressful environment and then check for the expression level of MIR155. So I don't know in human vascular smooth muscle cells, but I think it has been reported in other cells. Yeah.
Okay, thank you, Dr. Pang. I have a question for Ms. Norazira. Ms. Norazira, you are here, right? The question is, nice talk, Ms. Norazira. May I know why only male fetus and adolescents to study this miRNA and protein?
Okay, thank you for the question. Because it is actually BPA has been reported to have a sex-specific effect on the, for example, on the behavior outcomes. So according to the human prospective study, we can see from one study, the male one is more pronounced to have a lower scores in intelligence test than the girls. And then we have also animal study. The male rodents also commit a higher error in memory tasks than the female rats. That is the example from the behavior study. And then it may be because of female performance in neurobehavior is believed to be influenced by hormonal fluctuation. So that's why we go only for male. Because female, if we want to include the female, we need to have another groups. We cannot combine the results. So that's why we only focus on males. Thank you.
Okay, thank you, Ms. Norazira. Next question. There's another question for Ms. Norazira. May I know what study can be done to correlate the changes in the miRNA and protein with the function?
Sorry, may I take a time to
the question, it's in the chat. So what study can be done to correlate the changes in the miRNA and protein with the function?
Okay, I tried to answer the question. Actually, why I include the microRNA and a protein study in my research, because, for example, microRNA 19A and microRNA 539, they have a pattern of expression during fetus until adolescence. So that's why I include gestation day 21 and adolescence day 35. So the microRNA for my study is responsible to regulate the protein gluon N2A, MRI19A for the protein gluon N2A, MRI539 for protein gluon N2B. So that's why I correlate the microRNA and protein level together, and plus the changes of microRNA during the fetus, it showed that it can be inherited until adolescence. So that's why we have both ages in my study, which is the microRNA, also the microRNA and so the protein, because it's correlated to each other, the development. Am I answer the question?
Actually, because you say it's involved in the learning and memory function. So I just want to know, how would you correlate your finding to this?
Okay, sorry. Actually, we have done a neurobehavior test. So, but we have presented the results. So actually, according to our neurobehavior test, it showed that the BPA treated rats have impairment in the learning and memory task, which is when we see when there is a decrement in expression of microRNA and also protein. So we prove it by using neurobehavior test, but I didn't present the neurobehavior test here. But I can share to you that from the neurobehavior test, we have the proof that the BPA treated rats have a lower performance in the memory-related tasks. I think that's all.
Okay, thank you, Ms. Norazira. There is a question to Mr. Derek. Mr. Derek, the question is, did you make the LFD yourself? Has the detection limit of the system being tested? Mr. Derek, are you
I'm sorry, can you repeat the question?
Did you make the LFD yourself? Has the detection limit of the system being tested?
Okay, so the LFD itself, I did not make it myself. However, for the LFD, we designed the primers with specific labels that is meant for the LFD. So we can detect it on the LFD. But then for the detection limit, yeah, we have tested it together with the sensitivity test as presented earlier. It is also at the 100 copies.
Okay, so you have done the detection limit of the system, right? You have tested that.
Yes.
Okay, thank you, Mr. Derek. I just have one question for Dr. Faranas. you're looking at the cell migration. Is there any plan to look on the cell invasion? Do you any plan to do that? I would think it would be interesting to look at the invasion as well, since you have looked at the migration part.
Yeah, actually, this research has not been completed yet. And because some parts of the drug development and formulation is already completed, and we find that the effect after formulation, when we combine with a few carrier, was very promising. We are planning to extend the objective of this study. And I think, yeah, that's a good idea to look at the invasion, right? We are going to test it in animal model also.
Okay, that's very interesting. That will be interesting. And a quick question to Mr. Tan. Mr. Tan, you are here, right?
Yeah.
Yeah. You show us your results in the pathogenic and likely pathogenic variants just now in the DCM patients, right?
Correct.
Do you see this pattern, the same pattern in our neighboring countries, Southeast Asia? Do they have these variants as well? Or is only specific to our population, which your sample has showed these results?
Okay. If you actually look into the discussion part, so some of the variants like the pathogenics, like I have discussed, right, is to have some similar demographics. For example, about the NYBPC325 base pair deletion, the intronic variant is actually quite common in South Asian descents, like in India populations. So when we actually look back into our patient data, right, so it is also present in the Indian ethnicity, and also I think one is on Malay. So it's quite similar. So some actually have like similar with other cohorts as well lah. So there are also some unique ones. Yeah.
Okay. Thank you, Mr. Tan. My last question for Mr. Ashraf. I wish that you can show us your sushi just now, but it's not there. My question is, you show us that your results show a bit different from your hypothesis, where there are increase of DNA methylation. Do you think that it depends on the locus, or is it on the variants, because of the variants that you are seeing, or is it because, I don't know, locus variants that you are seeing the differences where high methylation, DNA methylations are reported in our populations?
Thank you for your question. Actually, we don't actually, we are not actually sure why is that, because in the other previous study, they were looking at around the similar region as well, but possibly at different CpG methylation site. So we don't actually know at the moment why is that, and because we don't have the data on expression, so this makes our findings somewhat incomplete. We couldn't manage to amplify the transcript, possibly because of it's very, very low level, as we know that it's mainly expressed in the brain. But some studies have also managed actually to measure in peripheral blood. So we tried to use their methods as well, but couldn't get an amplification for CSMD1. So we don't really sure. It's possibly because of other factors as well. You know, they have CSMD1 is also a target for microRNA, and it could also play a role. So yeah, it depends on context as well.
Okay. Thank you, Mr. Ashraf. I would like to thank all our speakers for today. Before we end our oral session, can I ask everyone to turn on your camera so that we can take a screenshot together before our closing ceremony? Okay, can I ask technical team to help? I think we have 5 pages here. Technical team, can you do the counting, please? Okay, I'm done for the page 1. I'm going to the second screen. 1, 2, 3. Third screen. 1, 2, 3. Okay, so thank you everyone, and for listening in today, and I pass to our MC of the day. We are going to have our closing and prize giving ceremony after.
Good morning, everyone. So the prize giving ceremony and closing ceremony will be starting at 11:25 a.m. Thank you.
Bioplex assay development is built on years of expertise with a rigorous SMARTS development approach that ensures best-in-class performance. Confidence in system performance is easy with the integration of the Bioplex 200 system, software, and consumables. Engage in the immunoassay workflow you're familiar with while getting results from multiple analytes in one well with less sample.
Ladies and gentlemen, good morning and welcome to the prize presentation and closing ceremony for the 5th International Conference on Molecular Biology and Biotechnology 2022. My name is Dr. Revati and I will be your MC. Before we begin, I would like to bring your attention to some of our housekeeping rules. Kindly keep your audio and video off throughout the event to avoid any disruptions and ensure a smooth running of the event. Please do turn on your video when instructed by the program committee. If you have left the room and would like to join again, kindly join by clicking Enter Hall 1 on the program in the welcome kit. Ladies and gentlemen, a feedback form has been shared in the chat box. Do take a few minutes to fill it. Please note that the e-certificates will be emailed to all the participants within 3 working weeks after the end of the conference. Let's move on to the exciting lucky draw session. The lucky draw session today will be streamed live by the ICMBB committee. There would be 4 prizes for today. And our first winner of the day is Ning Kwan Lam. And the second winner goes to Jonathan Han. The third lucky winner of the day is Narmani Murugan. And finally, for the last draw, the 4th lucky winner goes to Azwani Humaira Zakaria. Congratulations to all the winners. You will be contacted via email by the program committee at the end of the conference. Now, for the most awaited session of the day, the 5th International Conference on Molecular Biology and Biotechnology, ICMBB 2022 Awards Presentation Ceremony. Announcing the winners of the 5th ICMBB Best E-Poster Award. There are 3 winners in each of the respective streams, biotechnology and molecular biology. The winners of the 5th ICMBB 2022 Best E-Poster Award in the biotechnology category goes to Carissa Su Ann Wong from Monash University, Malaysia, Bao Chi Wong from Monash University, Malaysia, and Siti Liana Saud Ghani from University Kebangsaan, Malaysia. The winners of the 5th ICMBB 2022 Best E-Poster Award in the molecular biology category goes to Jennifer Kui Ling Chi from University Malaysia Sabah, Zain Nuraza from Malaysian Palm Oil Board, and Sophie KK Law from Monash University, Malaysia. Now, we will follow with special awards presented by the Malaysian Society of Molecular Biology and Biotechnology. The winners of the MSMBB STEM TikTok Challenge 2022 under the Best Content category is a team led by Miss Cheryl Nicole Hendricks from Management and Science University. The next winner is a team led by Nur Shifa Imanina binti Najri from University Putra, Malaysia. The winners for the most liked on Instagram are Vivashini Valentiran from Management and Science University and Rosina from University Science Malaysia. The two recipients for the MSMBB EduSTEMx Grant 2022 are Assistant Professor Dr. Tan Chun Hui from UCSI University, Mr. Muhammad Faisal bin Muhammad Bukhari from University Malaysia Sarawak. And finally, announcing the awards recipient of the MSMBB Best Scientist Award of the Year 2022 to two outstanding individuals. The Young Scientist Award 2022 goes to Associate Professor Dr. Crystal Lim Siu Ying from UCSI University. The Distinguished Scientist Award goes to Professor Dr. Jennifer N. Hari Krishna from University Malaya. Congratulations to all the 5th International Conference on Molecular Biology and Biotechnology ICMBB 2022 award winners. Now, we would like to invite the MSMBB Best Scientist Award winners to share a few words. Welcome, Associate Professor Dr. Crystal Lim Siu Ying.
Hello, Dr. Ravati, can you guys hear me?
Yes, Dr. Crystal, we can hear you.
All right, good morning, everyone, and thank you very much for this honor, for this award. And this means that this year I'm 40 years old, which you guys know that it's the last year for me to apply for this award. So I'd like to thank the organizing committee, MSMBB, the council, and the panel judges for this award. It is a very great honor, and thank you very much.
Thank you, Dr. Crystal, for the short and sweet talk. Now, let's welcome Professor Dr. Jennifer N. Hari Krishna to share a few words.
Thank you. First of all, I must say that I am very deeply honored to receive this award, and it means very much to me coming from the Society MSMBB, which has been alongside me throughout most of my career. Science is an unusual career. We're always looking for problems, and I wouldn't have been able to solve so many problems or learn and achieve so much without the amazing people who have inspired me, mentored me, collaborated with me, and supported me. Colleagues, postdocs, students, not only from UM, but also UPM and MUST, and many other research groups in Malaysia and overseas. And I especially want to thank my students who over the years and still today teach me a lot and give so much wonderful support and inspiration in the lab. Never lose your enthusiasm. Science is a never-ending journey. There's always more to learn and explore, and it's my privilege to work in this field with so many wonderful colleagues and people, and to have had such fantastic support from funding agencies and the institutions that have supported my career, and of course, including my beloved family and my MSMBB family among those institutions. Thank you very much.
Thank you, Professor Dr. Jennifer Ann. Without further ado, allow me to invite MSMBB President, Professor Dr. Lau Yiling, to deliver her closing address.
Hear me?
Yes, we can hear you, Prof.
Okay, I must first apologize that my video is not working, so you can't see me, but my face is the same as my profile picture. Congratulations to all the winners. Very, very well deserved. I'm truly happy for you guys. Dear conference chair, colleagues and friends, first and foremost, I would like to express my appreciation to the chair of 5th ICMBB, Dr. Damani Devi, for leading the conference and forming an excellent team. Thank you, Damani. And also thanks to all the heads of each subcommittee, including Dr. Wong Shok Mui, here, Dr. Wong Shok Mui, for your constant reminders and email communication with all of us. Thank you so much. Dr. Amira and also Dr. Lai for making sure that we are financially able to run the conference. Prof. Jamal and the scientific team, a very tough job. Thank you for screening through hundreds of submissions. Very well done. And Dr. Ellen and team for publicizing our conference. Very importantly, Dr. Arun for making sure the conference runs smoothly, and also his team, of course. And not to forget, Dr. Krista for a very successful pre-conference workshop. Very well done, everyone. Also, a big thank you to the keynote speakers, Professor Asma Ismail and Professor Lui Laiming, plenary and invited speakers, all the speakers for sharing your expertise and knowledge with us. We have learned so much from your presentation in the past 3 days. And not to forget, very special note of appreciation to our sponsors for their financial support, Cambio, NextGene, Promega, Bio3, Stanrose, Medigene, Expired, Trios, and Ideal Scientific. We truly appreciate your interest in supporting our conference. So I would also like to take this opportunity to briefly introduce the Malaysian Society for Molecular Biology and Biotechnology. So MSMBB is a non-profit organization with the objective of promoting communication among scientists in the field of molecular biology, cell biology, and biotechnology. As you know, we regularly organize conferences, seminars, workshops, and competitions such as MSMBB STEM TikTok Video Challenge and Best Scientist Awards. We also provide grant for community outreach programs. On top of that, we also manage a Scopus Index Journal, Asian Pacific Journal of Molecular Biology and Biotechnology, which I believe will be promoted to ISI Index soon. So why not join the coolest society in Malaysia? I hope it is, to enjoy special rates for our conference, seminars, and publications. Who knows, you may be the next MSMBB Best Scientist. Reach out to us by scanning this QR code or browsing through our social media. So finally, this is the end of our conference. On behalf of the organizing committee, I would like to express my heartfelt gratitude to all of you who participated in this conference. And I believe that good experiences have been shared and good lessons have been learned. I hope we'll meet again next year in 6th ICMBB. Bye and stay safe. Thank you. I'll pass the floor back to Revanti. Thank you.
Thank you, Professor Dr. Lau Yiling, for your insightful and inspirational words. Ladies and gentlemen, kindly do not forget to fill in the feedback survey that has been shared in the chat box. We now request everyone present to turn on your videos for a group photograph. Technical, can you please take the screenshot?
Yep. So on my count of 3, just keep smiling. I'm sorry you have to smile a little longer because there's many participants. I've got multiple pages here.
No worries, Arun, we understand. Just take a smile. Congrats, Prof. Jenny.
Okay. Thanks a lot for the smile. Have a great day ahead.
Congratulations to all the winners.
Thank you to all the participants. Thank you. See you all again. Bye.
See you all next year.
Next year, wonderful job, Damani. Yay.
Don't forget our AGM.
Thank you, everyone.
At 2 o'clock.
Yeah, not forgetting. She'll join later.
Before we end, a gentle reminder to all the MSMBB members that the 33rd MSMBB annual grand meeting will be held on 9th June 2022 this afternoon at 2 p.m. The link to join this AGM has been sent to your email. Do come and join us for the AGM. Also, please remember to fill up the feedback form for the conference. The link is provided in the chat. Thank you, everyone. With that, the ICMBB 2022 ends. See you again next year.
Bye, everyone.
Bye, Kanti. Bye, everyone.
Bye. Congrats, Jenny. Congrats, Crystal.
Bye, everyone.
Bye. Congrats, Prof. Jennifer and Crystal. Bye-bye.
Bye.
Bye, and thank you for that conference. Bye, thank you so much.